He got his PSc from Punjab Agriculture University from India, and his major was plant breeding and genetics, and especially in biotechnology. Then he moved to USA to get his PhD from Washington State University, USA, and his major also was plant breeding. Dr. Karansher published 15 international manuscripts in high international profile journals such as Plant Genome, Frontiers in Genetics, Frontiers in Plant Science, and so other many. He gets some prestigious awards. In 2021, Outstanding PhD Student Award from Association of Agricultural Scientists, Crop Science Graduate Students Award from Crop Science Society of America. He also get a very interesting award, the Prologue Scholar Award from National Association of Plant Breeders. Also another prestigious award, Grilad or Motto Award from Crop Science Society of America. And I mean, we have so many awards in his CV. So he is working as an editor in Journal of Crop Improvement, and he is review editors in Frontiers in Plant Science and Frontiers in Genetics. Thank you so much, Dr. Kancher, for accepting our invitation and present your remarkable research through MGBS. Today we have like the presentation title will be Genomic and Phenomic Selection in Plant Breeding Programs. Actually, this is very important and interesting lectures. Thank you, and please start.
Yeah, thanks a lot, Dr. Salam, for the great overview, and thanks a lot for the invitation. So hello, everyone. So myself, Karansher Sandhu, and currently I'm a product development scientist at Bayer Crop Science. And today I will be giving a presentation on all of my PhD work, the work which I did at Washington State University. And the title of my presentation is Genomic and Phenomic Selection in Plant Breeding Programs. So I will just turn off my video just to reduce the bandwidth issue. So if we look at this figure, so this figure shows the yield and the area harvested for 6 important crops which are grown worldwide. So here I'm showing the yield for crops like rice, wheat, soybean, common bean, chickpea, groundnut. So if we see the progress of improvement from 1960s to 2020, we can see that for certain crops, there was a rapid improvement in yield in 1970s to 1990s, like for crops like wheat, rice. But now the yield of the most of the crops is stagnated. So for example, if you look at the yield of common bean, chickpea, even wheat to some extent. So there is a plateau which is being reached. So we need more technologies or techniques so that we can break this barrier and can rapidly increase the genetic gain as well as grain yield in order to feed the increasing population. So we need another kind of a green revolution or a rapid increment in the grain yield for the different crops. And on the right hand side, you can see the area from which these crops are harvested. So there is no increment of the area from which these crops are being harvested. That means we have the same land to produce the more population, suggesting we need more per unit land production to increase that. The only crop for which the area has been increased has been soybean. That can be associated to its high protein requirement. So soybean can increase its area, but at the cost of other crops. So we need to have a balance which can only be achieved by increasing the yield of the different crops. So I did the similar kind of analysis looking at the within continent variation. So here I'm showing the yield of, again, those 6 crops. Do I'm audible? I just saw a chat that I'm not audible.
We can hear you, I think.
Oh, okay, that's fine. Okay. So then we did the same kind of analysis to look at the within the continent variation. So sometimes some continents are performing better, like others may not be. It can be attributed to the germplasm or the genetic efforts which those breeding programs are doing, as well as the environmental condition. So if we look at only for the wheat, so Europe and North America, the yield of wheat is increasing more compared to the countries like in Asia as well as Australia. So, but wheat is mostly consumed in Asia. So we need to do more effort so that we can increase the yield of wheat at a more rapid pace. So similar kinds of trends we observe for the different crops. Like for example, if we have to look at the common bean. So again, North America, the yield of common bean is way more than the other countries like in Africa, where it is consumed way more often than the other countries in the world. So we observe the similar kind of the patterns within the different continents, like some continents are performing better in regard to particular crop, which is due to their funding agency. So some fund agencies are giving more funds for a specific crop in a specific region. So we need to divert those efforts in a such a way that there is a equivalent improvement of all those crops at the different continents so that we can increase the yield in a long run. So this is a barcode for the manuscript from which I'm talking about, like where we did this all the analysis and to show how we can improve the genetic gain in the different crops. So as in a breeder's life, these are the three main things which they perform. So first thing breeder has to do is crossing. So in order to do crossing, they need to decide the best parents to cross. So think they can consider different things like how much genetic variation they have pooled. Do they want to cross best versus best, or the different things which they can use to decide those crosses. So these days the most commonly used is the genomic selection. So based on the genomic selection, you can decide, okay, these two parents can produce such a good progeny. So let's decide to make those crosses. So once the crosses are done, then the other part of breeder's life is to perform evaluation. So during the early stage in the pipeline, we have a large number of lines, so lines in a thousand. So we perform selections from like highly heritable traits like some of the agronomic or the disease traits. So after that, we perform selections like yield trial evaluation. So at this time, we have our lines which are disease resistance, have good agronomic traits, and are fitting to your environmental conditions. So then we will evaluate them for the different yield attributes. And the final and the most important phase is a selection. So this curve just shows a normal distribution for any of the traits. As a breeder, our main goal is to select the top 10% or the top 5%. So whatever the lines you want to select, so based on that, we make the selection. Depending upon how you are making selection, that's how we can calculate the genetic gain. For example, if the previous yield was 2 ton per hectare and you are able to improve it to 3 ton per hectare, that means you achieved a 1 ton per hectare genetic gain. So that is our ultimate goal, to move in a positive direction, to keep continuously making progress in a breeding program. So genetic gain, so as I mentioned that thing couple of times, so genetic gain is the most important things in any breeding program to measure the success. So like we measure the success of a student with their marks, how they're performing in their classes. But for a plant breeder, we make our estimation based on the genetic gain. So this is the most commonly used genetic gain equation. And from this, you can see that genetic gain is affected by four most important components. So things like selection intensity. So selection intensity means what is the percentage of lines you selected during your breeding program. So you don't want to select like 20% or 50% because we have the limited resources. So it plays a very important factor. What is your selection intensity in a breeding program? And the second factor is genetic variance. So you also need to consider that there is enough variance available in your germplasm so that you are not continuously moving into one direction. So enough variation should be available so that you can continuously keep making selection. And the third factor is selection accuracy, or you can call it heritability. So like for the traits like disease resistance or certain other traits for which we have the marker, we can say that the heritability of a trait is 100%. That means if a plant is having that specific marker, that gene will be present. But for the trait like grain yield or some other quantitative trait, the heritability is usually less. So we need to consider those aspects again into the genetic gain equation. And the last and the most important factor is year per cycle. So how many years you take from the day you made a cross to the day you release that variety. So typically in a breeding program, it usually takes anywhere between 8 to 10 years. But if we can reduce the year per cycle by using techniques like genomic prediction, phenomic selection, speed breeding, or genome editing, so it will be easy to manipulate the year per cycle so that we can release our varieties in 5 to 7 years by using things like rapid generation advance under the controlled condition. So right now, the main focus of most of the people is to reduce the year per cycle so that we can launch the product early in the markets for the farmers. So in order to get more information, you can go and refer to this manuscript. So now I will give a little bit context about genomic prediction. So as I mentioned a lot about genomic prediction, so what that technique is. So I assume all of you are familiar with techniques like quantitative QTL mapping, GWAS. So this thing is exactly like that. So genomic prediction is a technique which uses the genome-wide marker. So it will use all the marker information present in a plant to predict the performances of an individual. So for example, if you have a seed or a tissue, a plant which is just planted, like it's been 1 week, you can go to the field, extract the DNA and get all the genotypic information and then put all the information in our model. And the model can tell you the performance of a plant. It can tell, okay, this plant will yield 100 tons per hectare, or it will be resistant to a particular disease. So whatever characteristic you want, you can get with the help of a DNA information for that plant. So in order to do so, we need to train our model. So model training can be performed by previous years of a data set. So in our breeding program, we usually have a lot of genetic as well as phenotypic information from previous year data set. So we can get all that DNA as well as phenotypic information to train our model. So once you have trained your model, so in the next year, you just need the DNA or the genotypic information for those lines. Put all that information into your model, which will take a couple of days, and then the model will predict the performances of all the individuals. And based on that genetic estimated breeding values from the model, which we call GEBV values, you can make the selections. Okay, these are the plants which I want to go into the field condition. So with the help of genomic prediction, you can decrease the year per cycle, so the number of years which are required. You can increase the selection accuracy. So it's not like you are evaluating under the field condition where you have a lot of environmental influence. So you can increase that. You can also increase the selection intensity, because when we have a plot, we have a limited area where we can grow all of those things. But with the help of genomic selection, you can evaluate as many plants as you want, because the cost of genotyping is reducing day by day. So that way we can evaluate more plants. So from these techniques, we can clearly show that with the help of genomic prediction or genomic selection, we can rapidly increase our genetic gains in the breeding program. So here I'm just showing a trend of the publications which were mentioning the genomic selection. So again, I'm showing the trends for those 6 crops like rice, wheat, soybean, chickpea, common bean, and groundnut. So here I went to PubMed and searched in the abstract of the title the word genomic selection and the specific crop, just to see what are the different crops which are adopting genomic selection at a rapid pace and what's the reason behind those. So if you see the number of publications which are using genomic selection from 2011 to 2020, so wheat is the leading. So many researchers in North America as well as Europe and Australia, they are using the genomic selection to decide their parents, advance their progenies, as well as make the selection decisions, or even some researchers are using the genomic selection to release the hybrid wheat. So genomic selection is being rapidly adopted in rice and wheat, but for other crops like soybean, chickpea, common bean, groundnut, this researchers are still adopting, they are building their training models, so more progress is needed in certain crops. So now I will just give you an overview about physiological assisted breeding, or like how we can use the phenomic or high throughput data set in our breeding program. So if you look at this image, so this shows the electromagnetic spectrum of a plant. So electromagnetic spectrum from visible as well as near infrared region. So region from 400 to 2000 nanometers. So in the visible region, we can get information about chlorophyll content of a plant or certain kind of a leaf pigment. While when we are measuring reflection in the near infrared region, we can get information about cell structure, what are the different stress plant is having. And same when we are measuring reflection in the shortwave infrared region, we can get information about water content, leaf pigment. So all these things can be achieved by using different kind of a cameras like RGB or the multispect camera, hyperspect camera. So with the help of these cameras, we can get a tremendous amount of information from the plant, and all that information can be further used for making selection for low heritable traits like grain yield. So we know in grain yield there is the effect of genetic component, but it's not a lot. So we need more components to reduce the environmental variation. So all these physiological traits play a very important role if you're breeding for stress tolerance or some kind of a drought stress. So these traits play a very important role. And in one of my studies, so we extracted couple of different vegetation indices. The indices like NDVI, water index, simple ratio, GNDVI, so a couple more. So all these indices provide the different information like plant health, plant water stress, chlorophyll content, anthocyanin pigment. So with the help of all the reflectance which we measure from the plant, with the help of different sensors or which are equipped on drone or the satellite imagery, we can get a lot of information about plant. And all these things can be added into our genomic prediction model to improve the performances of the model. And all those things are evaluated into the multi-trait model. So previously, I just mentioned the genomic prediction, but in genomic prediction models, we can predict multiple traits simultaneously by using multi-trait model. So we can predict gain yield by using all these traits into our model to make more selection more accurately. So these things help us to improve our genomic prediction models. So just to give an overview about high throughput phenotyping, for how it's getting so common and what are the different platforms which are available. So mostly it can be performed by aerial phenotyping by using the tools like UAV, which we call drone, or by using remote sensing. So there are so many satellites which are revolving around the Earth. So all the data sets are freely available. So we can download those data sets by using the different QGIS. So then all those things have different kind of a cameras. So as I pointed out earlier, so cameras like RGB, multispec, hyperspec, thermal, and NIR imagery. So all these cameras can be used to measure different kinds of traits from a plant. So these cameras provide us information about different diseases or a pest. So whenever a plant is experiencing certain kind of a disease pressure, their leaves will be discolored. So RGB cameras can tell you, okay, there is some disease in the particular plot, so we need to take certain action. Similarly, some of the physiological traits can tell us, okay, something is bad is happening with this specific plot. We need to make some treatment or adjustment. So with the help of these sensors, we can look as abiotic, biotic, as well as different yield related traits. So the field of high throughput phenotyping is getting very common in the different breeding program. When you're screening the large number of lines, you don't have enough genetic variation, but with the help of phenomic variation, we can make selections and move our genetic gain in the one direction continuously. So similar kind of research I did for the phenomics, like what are the different crops and how often they are using high throughput phenotyping into their breeding program. So again, same kind of a trend we observe, wheat and rice are leading. So they are performing more studies based on high throughput phenotyping to make selections in any kind of a breeding program. So this figure is very important. So if you talk about a breeding program, so in a breeding program, we start with a new candidate. We decide the candidate or the parents. Then the crosses are made, and those crosses need to be made inbred or like completely homozygous by using things like double haploid production, speed breeding, rapid generation, advanced, or like single seed descent. So those things can be performed in a 1 year, like if you're using the greenhouse condition. And then we perform early stage testing, and then we perform preliminary yield trial, advanced yield trial, elite yield trial, and at the end of 8 years, we will be able to release the variety. That is a typical breeding program which is not using a lot of these new techniques, but we can integrate things like genomic selection here. high throughput phenotyping here that will reduce the number of years by 1 or 2 years minimum. That way we can affect our genetic gain. So for example, if you have run this thing in your breeding program, you have enough data set to train your genomic prediction model. So based on the previous year data set, for the next year, you can predict the performances of the F2 individual, or you can predict the performances of F3 or F4 lines based on the previous year data set. With the help of that, you can reduce the number of years, so you don't have to evaluate the yield on the EYT trial or the preliminary yield trial. So with the help of that, you can skip one of this trial. You can also predict the performances of the parents, okay, which parents we should use to make crosses. So all these things can be achieved with the help of a genomic prediction. And the other thing you can include in your breeding program is high throughput phenotyping. So during the late stage testing in a pipeline, we can collect the phenomic data by using the different kind of a sensor. And all these sensors provide us a different kind of information which can aid us in making more efficient selection. So this is a typical pipeline which is used in the most breeding program. But with the help of integration of things like genomic prediction, phenomic selection, rapid generation, advanced speed breeding, we will be able to reduce the number of years to 6 to release a particular cultivar in a breeding program. So just to give an overview, so this is the thing which different breeding programs are trying or adjusting their breeding program to accumulate this. So now I will talk about some few minutes for a study which we did in wheat. So the study was titled Deep Learning for Predicting Complex Traits in a Spring Wheat Breeding Program. So here, in spite of using traditional statistical model for predicting the different traits, we optimize the different deep learning models. So deep learning is like the things which are used in the different computer science branches, how the model can learn all the information and then can make predictions. So similar things are used in our iPhone, like when they recognize our face and then they open our mobile app. So same things we try to use in our study, like how we can use the previous year data sets to make the predictions for future. So deep learning models behave as the biological neurons behave. So in our brain, we think different things, okay, how these things can do a particular task or whatever. We make different kind of a assumption. So in a similar way, deep learning model consists of a set of neurons. So all those neurons interact with each other to make the predictions. So the main issue with the deep learning model is the training or the parameter optimization is a problem. So we need to consider different kind of a technique to optimize these deep learning models. So the best solution is you need to have some backgrounds in the machine learning or the computer science branch, or you can collaborate with someone who has these kind of a skill set so that they can build model for you based on your data set. So in this study, we evaluated two of the deep learning model. So first one is the MLP model, which stands for multi-layer perceptron, and the second one was convolutional neural network, which stands for CNN. So just to give an overview about the MLP model, so just to sake of time, so this model consists of an input layer. So in input layer, we provided all the genotypic information. So in this case, we provided all the SNP data to our model. And then it consists of different number of hidden layers. So you need to optimize how many hidden layers you want in your model. And each hidden layer consists of different neurons. So all these neurons interact with each other, make different kind of assumption or learn the pattern from the data to make the prediction for our output. So once you have this model trained, so in the next year, you just need to put the genotyping information or the SNP data, and that model will predict the phenotypic response of any individual. So once you have a model trained, so the training model is a cumbersome or the tiring process, but once you have everything trained, your model will be good for a couple of years. You can still keep updating those things, but it can make some accurate prediction. So similar kind of a thing is the CNN model, which we call convolutional neural network, but it consists of different kind of layers like convolutional layer, max pool layer. So, but the principle is exactly same. So data is provided to predict the phenotypic response at the end. So just to give an overview about this study. So for this study, we use the data set from our wheat breeding program at Washington State University, and the whole population was sequenced by using different genotyping techniques like GBS as well as 90K SNP chip. And the data analysis was performed by three models. So MLP and CNN were our deep learning model, and we compared the performances with the RR-BLUP model. So RR-BLUP is the most often used genomic prediction model. So we want to see, is there any advantage of using deep learning model compared to the traditionally or the statistically used model, which was RR-BLUP model? And we use different things like these are the computer science or the machine learning terms. So things like grid search, cross validation, early stopping, dropout, or regularization. So all these things were added into our code so that we can avoid the overfitting of our model. And these were the some of the libraries or the packages which were used for performing this study. And all the codes which were built in this study were available on the GitHub site. So you can go to the manuscript, use all the codes, and train your model so that you can make predictions in your breeding program. So for this study, we predicted the 5 different traits. So the traits like grain yield, grain protein content, test weight, plant height, and heading date. So here you can look at the heritability value for those 5 traits for the 3 environments for which this population was evaluated. So as we are all familiar that grain yield has a less heritability value compared to the other traits, so we observe the same kind of a trend in our breeding program. And then we optimized our deep learning model. So here I'm showing the result for MLP model, while here I'm showing the result for the CNN model. So we were able to optimize that set of different hyperparameters. So these are the different hyperparameters combinations which were tried into our models to make predictions. So we concluded that each trait requires a specific combination of hyperparameters to give us the maximum prediction accuracy. So with the help of this study, we were able to predict traits for the most important traits in our breeding program. Furthermore, we were able to evaluate or optimize the hyperparameters which should be used in a wheat breeding program. So just talking about the results. So in genomic prediction, so the results are usually achieved in the prediction accuracy. So it can vary anywhere between 0 to 100 or 0 to 1. So here I'm showing the result from 0 to 1 category. So you can multiply it by 100 and we can say, okay, our model was like 60% accurate or 50% accurate. So the first graph is for the RR-BLUP, while the green color and the blue colors are for the deep learning model. And we observed that for the grain yield, the deep learning models were performing 0 to 5% better compared to the RR-BLUP based model. And similar kind of a result we observed for all other 4 traits. So with the help of this study, we were able to conclude that there is a potential of using deep learning models if we want to improve the prediction accuracy of our model. And these models can be further improved by using more data into our model or optimizing those things in a certain way so that we can improve our prediction accuracies further. So we performed a similar kind of a study like how many markers are required into our model. So as I mentioned, we need marker as well as phenotypic information to train our model. So we did the same kind of a study like how many markers are required to predict the grain yield, but with the help of different models. Similarly for the grain protein content, test weight, plant height, and days to heading. So we observed that different traits require a different number of markers to achieve the highest prediction accuracy. Like for example, if you look at the plant height, so if for plant height, even the 1000 markers is enough to get the highest prediction accuracy. So after that, whatever the number of markers you keep providing to the model, there is no significant improvement of prediction accuracy. But for traits like grain protein content, we'll need a little bit more marker number which are uniformly distributed over the genome. So in this way, we were able to optimize like what are the different number of markers which are required for a particular trait to predict to give us a maximum prediction accuracy. So now moving to the last phase of this talk. So far we have talked how we can use the genomic information, how we can use the machine learning into our breeding program to predict the particular trait. But now I will talk about how we can integrate these 3 things like genomic data, data from a high throughput phenotyping platforms like UAV or any kind of a imaging sensors into our machine learning model so that we can predict the different traits into our breeding program. So these are the 3 studies. So I will be sharing a couple of results from these 3 studies. So this was our first study where we thought, okay, how about combining genomic and phenomic information into our breeding program? And this is our next one where we use machine learning. Okay, how could you use machine learning, genomic information, and phenomic data into our breeding program so that we can predict all the traits with a better accuracy? So again, for this study, so the spectral data was collected with the different sensors like UAV as well as handheld sensor. So then with the help of this sensor, we extracted in number of vegetation indices like NDVI, water stress index, anthocyanin stress index. So various indices were extracted with the help of all the spectral band. And these were the some of the R packages which were used for pre-processing of this data. So we need to filter data, impute couple of things. So these were the some of the packages which were used in this study. And we used four different kind of a model. So first one is univariate genomic selection model. So what this means, we are just using the genetic information to make predictions for a particular trait, for example, grain yield. So here we just use the genomic information for prediction. So in the second case, which we call SRI only model, so SRI stands for spectral reflectance indices. We only use the data which we collected with the help of a drone. So all the phenotypic data set with the help of a drone which we collected was used to train our models to predict the grain yield. So in the third scenario, we use grain genomic as well as spectral data as a fixed effect. So here we thought, okay, let's remove all the variation which our spectral data can explain and then use the genetic information for making prediction. So this was the third scenario. And the fourth was the multi-trait scenario. So multi-trait means we are predicting multiple traits simultaneously, like grain yield, grain protein content, spectral information. So all that information is integrated into the multi-trait model. So multi-trait model try to improve the prediction when a trait has low heritable value and the different traits are correlated with each other. So that was our ultimate goal, to integrate all the information into our models and to check what is the improvement of prediction accuracy. So here again, the performances for grain yield and grain protein content with the four versions of model which I just explained. So uni means only genetic information, SRI means only UAV data was used or the spectral data, and the third scenario where we included SRI data as a fixed effect, and the multi-trait means we are predicting multiple traits simultaneously. So if you look at this table, so the prediction accuracy for grain yield was 43% when we just used the genetic information, while it improved to 57 or the 58%. So you can see we can improve the prediction accuracy around 20 to 25%. So if we look at the ratio, so with the help of integrating the spectral or the phenomic, genomic, machine learning, so when we integrate all those things, we can improve the model performances by 20 to 25%, which is a lot if you are running your breeding program. So small increment in prediction accuracy will have a larger effect when you are making certain kind of a selection. And we observe a similar kind of a trend for grain protein content and for the different traits for which this population was evaluated, that when we are combining genomic, phenomic, and the machine learning models for our traits, we are getting improvement of prediction accuracy. So now I'm just showing the result for grain protein content. So here we added individual SRI into each model. So here we added all the information what was available, but here we thought, how about adding one parameter at a time into our model? So the first scenario is only using the genetic information. We are not adding anything else than the genetic information. While in the other scenario, we are adding one trait at a time. So here we are adding anthocyanin reflectance indices or GNDVI, which stands for green normalized different vegetation indexes. So we tried different indices and we concluded that for grain protein content, GNDVI is the most important parameter to improve the prediction accuracy. That is because GNDVI measure reflection around 550 nanometer of the electromagnetic spectra, which is indirectly linked to the nitrogen content of a plant. And we know grain protein content is equivalent to how much nitrogen is present so that it can be used for the synthesis of amino acid and ultimately for the protein. So in an indirect way, GNDVI provides us an estimate of grain protein content. and we can integrate the GNDVI into our genomic prediction model to improve the prediction accuracy from 50 to 0.65%. So with the help of this study, we were able to conclude that GNDVI is most important index if we want to predict the grain protein content. And we did the similar kind of a study for grain yield, but we concluded that for grain yield, we need all these indices to improve the prediction accuracy. That is because grain yield is a complex trait, so all these traits provide information about different physiological traits and ultimately help us to improve the prediction accuracy. So this is the other scenario which we tried was the unit-trait and the multi-trait scenario. So again, unit-trait means only one trait was predicted, multi-trait means multiple traits were predicted. So as you can see, all the versions, so whenever we are using a multi-trait model, there is the improvement of prediction accuracy for each of the model. And we also evaluated 9 different models in this study. So first was the G-blub or the RR-blub. Then we evaluated a couple of Bayesian models. So Bayesian models are very popular in the genomic prediction. Then we evaluated 2 machine learning models, which were like random forest and support vector machine. And finally, we evaluated 2 deep learning models, MLP and CNN. And we concluded that random forest, which is one of the machine learning model, or the MLP, which is one of the deep learning model, they perform better compared to all other scenarios. So we concluded that we can use either random forest or MLP model in our breeding program if we have enough data sets in the breeding program so that we can improve the prediction accuracy. So this is the last figure. So this shows that how we can use the previous year data set. So here we trained our model on 2014 data sets to make prediction for 2015. Similar kind of results were performed by using data from 2014 to make prediction for 16, or training data for 2015 to make prediction for 2016. And with the same kind of analysis for grain yield, grain protein content, and for all these scenarios. And you can continuously update your model every year. So 1 year you collected the phenotypic data set, include everything into your model so that you can make predictions for the future years. And with the help of this study, we found that there is a potential of using genomic prediction, phenomic selection into our breeding program so that we can train our model, make predictions more earlier, more accurately into our breeding program and make selections or advancement at a rapid pace. So with this, I would like to thank all of my PhD committee members and mostly my advisor, Dr. Aaron Carter at WSU. So with whose help I was able to do all this research. And thanks, Dr. Salam, again for the invitation. And I will be happy to take any questions. And if you guys have any question after this talk, so feel free to send me a message on LinkedIn or Twitter. So you can just scan this barcode and send me any questions if you guys have. So, and I'm happy to take questions. Thanks for listening.
Thank you so much, Dr. Cannon, for really very interesting lectures. I mean, all of us learn a lot about genomic selection and prediction. And we have a question here
Yeah.
from, yeah, please go ahead. I don't know, just nicknames. Uh, yeah, your mic now is allowed. You can just speak. Bushra, I think. Bushra Fordil. Please unmute your mic. Yes.
Hello, sir.
Yeah,
So
hi.
I have a question.
Yes, yeah.
So you talk about a number of softwares. So where we can find these software easily available to predict our data in our laboratory? Are these available online, or so what are the sources, how we can get them?
Yeah, so all those things are available on the different platforms. So if you go to any of the publications which they are using these models, so they make their codes available publicly on a platform which we call GitHub. So the main requirement is you need to know at least one of the language like R or Python so that you can tweak or modify that code according to your requirement. But all these things are publicly available on a platform which we call GitHub. So GitHub is used to share all these codes publicly across the different reading program.
Okay, sir, can you spell that?
Yeah, I can type in the chat. Yeah, sure. But the thing is like you need to go to the different papers. So you can go to one of my paper and find that code which is available on the GitHub. So that way you will be able to find all those things. Yeah.
Okay, sir. Thank you so much. Thank you.
Yeah, sure. Yeah.
So any question? I think maybe go to chat if yeah, I think there is a question in from Sumaya Hassan. Do we get access to the recording? Yeah, okay. This is uh, yeah, maybe we will we can post our channel, MGBS channel. Just we need some days to upload the videos. So just you can subscribe the channel, and when it becomes available, will get a notification. I think one of assistant, please put the link of a channel on the chat room. So any questions? Okay. Yeah, from Asad Razak. Sorry, Dr. Yasser Morsi. Go ahead, please.
Uh, I'd like to thank the organization committee, and I'd like to thank Dr. Karansher for this very nice and informative lecture. I do have two questions. The first one, did you give all markers the same weight as the training model?
Oh, okay. So the thing is like we don't give the same weightage to all the markers. Like we know some genetic regions have more effect by compared to the other. So model do these things. So when I talk about we use machine learning model or we use the Bayesian model. So all these model have different kind of a features. So Bayesian model, they say all markers have equal weightage, while RR Blub say opposite, machine learning models say opposite. So different models have their different assumptions. So some models say all markers have equal while others. So you can make those manipulations into the models to adjust according to your requirement.
Okay, thank you. The second
Yeah.
question is, um, can we identify the markers that are associated with the traits of interest using genomic selection? Because sometimes we cannot genotype all the germplasm do we have, and if we can identify the markers that are associated with the trait of interest, we can use it to select for, yeah, in another germplasm, not the genotyped one.
Yeah, that's a really good question. So as we are aware, usually we use techniques like GBS or 90K, any kind of a SNP platforms, and those things are pretty expensive in a breeding program. So now a new techniques is being involved, which developed, which is called skim sequencing. So skim sequencing will involve only 1,000 marker. So that means whichever trait you are interested in, that chip will only have 1,000 marker for your trait. So you just need that information, like as you mentioned we can identify all those markers. So different peoples have done the research and identified, okay, these are only the genomic regions which are associated with green yield. So we just need to genotype these regions. So research is being done and hopefully that platform will be available to all the researchers within a year or two so that only that information can be used.
Yeah, thank you, Dr. Karansher. Thank you.
Yeah. Yeah,
Uh,
thanks.
yeah, we have next in line, Asad Razak. Please go ahead.
Yeah, Asad Riaz. Yeah. Hi, Karan. First of all, I would like to thanks to you and Dr. Ahmed. Uh, Actually, I don't have any background about programming or any other language like R or Python you mentioned before.
Yeah.
Is there any option or like your recommendation where we can learn about how to train our models or how to develop the models?
So yeah, I can share some of the courses which are available online. So the thing is like you might have to spend couple of weeks to understand what are these model and how to program and so that you can tweak or make changes into the public available codes. So it will be a couple of weeks
Yeah,
effort, but once you do that, it will be good for remaining 20-30 years the way you're doing the research. So you might have to put certain effort on only on the one of the language. If you can spend a couple of days on R, you should be able to good to make all these changes into the codes. And I can share some of the
yeah,
courses which are available online, which you can do or I did during my PhD.
Okay, that will be really grateful.
Yeah, sure.
And the next thing is about some of the traits you mentioned in your research. So these days, what type of traits or what crops you are focusing in your research, or what job you are doing in where you have mentioned here in the slide, like.
Mm. So right now I'm a soybean product development scientist at Bayer. Regarding the work which I'm doing, so that is a company policy, so I won't be able to tell much about that, what I'm doing there. But it's a soybean breeding, and we are kind of using similar kind of a techniques, but in a other way around. Yeah.
Yeah. Okay.
Mm. Yeah.
Actually, the reason why I asked this question, I am interested to work on barley shoot development and sorghum shoot development. It's
Okay.
a combined project, you can say, with the Queensland University professor David Jordan and uh, one professor from China, Zhejiang University. So that's why I was asking, like, do you know or have any experience about using these models on the trait or these practices on the trait of shoot branching or like shoot
Yeah.
development?
Yeah, that you can use. So yeah, we have also tried that thing in wheat. So one of my colleague at in my WSU, so he tried all of those things on the shoot characteristics. Like in hybrid wheat, we need to consider different things like florets and spikelet traits. So we did that
Mm.
thing as well, and we also did for the quality traits in wheat. So you can implement this model on any kind of a trait. So as you're trying on the different shoot traits, so it should work perfectly on this as well. Yeah.
Okay, that's all. Thank you so much. And I will be waiting for your links about the uh, tutorials and the videos,
Yeah, sure.
like
I will
direct
send you. I will
courses.
send you after the meeting.
Okay, thank you so much. Thank you so much.
Thank you, Asad. Next in line, we have a question from uh, Vijaya Kumar. So please go ahead, unmute your mic.
Hello,
Yeah.
am I
Hi.
audible?
Yes.
Hi, Karan. I'm Vijay from ICSAT.
Hi.
Okay, I have one doubt. When
Yes.
we are considering the phenotypic data, we need to consider per se performance of those parental lines, or we need to cross those parental lines with some testers, and we need to take the phenotypic data of those hybrids. And is this phenotypic data collection, is this vary from self-pollinated crops and cross-pollinated crops? Is this any difference is there while collecting the phenotypic data?
No, it should be the exactly same, but the only thing is like you need to add different parameters into the model if you're doing it for a hybrid crop or if you're doing the self-pollinator crops. The only things you need to put into the models, other than the data collection should not be anything different. Yeah, it should be the normal data which you do collect in your daily routine.
Okay, so when we want to select the parental lines, so we need to take the phenotypic data of parental lines only.
Yes, only the parents' data and their ancestral lines, okay, how their ancestor perform under the particular conditions, and then you can predict their progeny performance.
Okay, like in some CIMMET papers, I have observed like they have crossed those lines with two testers. Then they have collected phenotypic data of those progeny, like parent hybrids. Then they used in that models. So I got that some confusion in that.
No, so you can try the both approaches. So nothing is wrong with both. So different people tries the different approach. The one which I told will save you couple of years. You don't have to evaluate them into the field conditions. So, but
Mhm.
with the CEMIT approach which you mentioned, you need to evaluate 1 year and then make this decisions.
Yeah, in my case, I'm using GBLUP by using
Yeah.
SOMAR. So in my case, I have used hybrid data only, hybrid
Yes.
phenotypic data and parental and genotypic data I have used.
Yeah. I think that's a good
Okay,
technique. Yeah.
okay, thank you.
Yeah. Sure.
Thank you. I think we have some question in the chat room. Yeah, how we can get contact to doctor to join work? Yeah, I think he in the poster that we sent, you can find the email address and just you can contact him
Yeah.
by email, asking for collaboration. Amira Murad, thank you for your interesting talk. Is there available machine that is specific for disease measuring?
Is there available machine that is specific for disease measuring? So I didn't understood your question. So what machine you are asking?
So means there are, I won't say machines, there are drones available. So just fly a drone over your field and drone can tell you, okay, your field is affected by leaf rust or is affected by powdery mildew. But I don't know any machine as such, but drones can do all those work.
Okay. I have a question for myself.
Yeah.
Um, you said about genomic selection and genomic prediction. Is the
Yes.
same or there is a slight difference between them?
Yeah, that both terms are interchangeably used. So genomic prediction, you can say, okay, we are training our model, but when we call genomic selection, we can talk about the breeding program. Okay, our program is based on a genomic selection approach. But when you train the model, so the most commonly used term is the genomic prediction, but there's nothing different like anything as such. Yeah. Different group use different way.
Yeah, okay. Another question regarding to the
Yeah.
selection gain equations that you
Yeah.
described. Um, you divided by years.
Yes.
And what about if we have like locations, different locations, and if we have location and years? And do you think that the climate change may affect the selection because the weather it changes from year by year, and we we predict or based on some years that maybe in the future we will have like warmer years or colder years.
So yeah, so as you mentioned, so we evaluate at the multiple location as well as multiple years. So that one, so when you say years, so y will be still the same at the denominator of that equation. But when you're evaluating a particular line at the more location, so the thing which will be affected is the r, so the accuracy component in that equation. So if you have a trial at multiple location, you will be more confident in your data set, and that will increase the selection accuracy and ultimately the genetic gain. But the year wise, it will still remain the same. So if you have trial at 5 locations in 1 year, so year is still one, but the R will increase based on your confidence on your data sets. So I hope that answer your
And
question. Yeah.
in your presentation that as we have seen that you predict with many traits like
Yeah.
green yield and protein component, but what about to select the genotypes in the testing populations? Because the testing population you genotype, but you don't phenotype. So have you really selected based on these models
Yeah.
um, perfect genotypes, and you, do you test them in the field? So just to make sure that you get the target.
Yeah, still we are testing those lines into the field conditions because we are not 100% confidence yet on these techniques. But as when we test those into the field condition and then we look at our model's results, okay, our model say this variety will be the 100% the first one, but that might be the second one, that might be the third one, but it's not like our model say this line will be the first or the top performing and that won't be the last. So still there are some problems we are improving into our models, but we are not reliant on this technique 100% yet, and not even mostly breeding programs. So all the breeding programs are still doing the field evaluation to some extent, but we are reducing like the poor performing lines. You can easily say, okay, these 50% line, the bottom 50% line won't perform good, so don't evaluate them into the field. But to decide the top of ones, you need the field data. Yeah.
Okay, we have one question in the chat room. So, hello, sir. How exactly you selected different numbers of markers to check the predictive ability of different models? Is it randomly or uniformly distributed markers across genome?
Yeah, so we use the GBS data and that was not that uniformly distributed. So as I mentioned, so new techniques have been developed like skim sequencing, which will be uniformly distributed. But as you provide like couple of thousands marker to the model randomly, so that works perfectly. So they automatically adapt to those markers conditions. Yeah. But if you provide on your own, okay, one marker near the telomeric region, one from the centromere, that will affect, but just provide all the information to the model and they will adjust those calculations.
Okay, thanks for answering. Do we have other questions or?
Yes, Dr. Salam, shall I ask
Yeah, please
one
go
more
ahead.
question?
Yes.
Mm, Um, I just would like to ask if we have two parents that they are contrasting for certain trait, and then we have them genotyped, can we produce the expected allele combination without having a progeny? And then from this allele combination, can we predict the performance of the genotype that will have this combination?
Yeah. So in 2018, so one group from University of Minnesota, so they released a R package. So doing the exactly same thing which you asked. So they said, okay, these are our two of the parents, and they simulated their progeny. Okay, if our progeny has this specific allele set, what will be their performances? So yeah, they have done, I don't remember who was the main author on that, but it was somewhere from University of Minnesota, and you can search, okay, our simulation package to simulate the progeny performances, and they have done this thing, and then you can use the same approach in your study, but we haven't tried that yet. Yeah.
Yes. I just would like to add something more. And based on this allele combination, if can we use based on this simulated data, can we induce this allele combination using something like genome editing or something else?
Yes, we can induce those things. Yeah, is it possible? But I don't know. We haven't tried it so far. Yeah.
Thank you.
Yeah.
Thank you, Dr. Yasser. Okay, do we have other questions? I think I think no further questions. Okay. Thank you again, Dr. Karin, share for your patient because you got a lot of questions this lecture. And thank you for the audience. And hope that you enjoyed with the lecture and the information, very useful information that is given by Dr. Karansher this seminar. And see you next week for new MGBS lectures. And for the recording video, I will speak with Dr. Karansher if he will allow us to publishing his video or recording, and maybe after um maybe few days to week we will publish it in our YouTube channel. I think the link was or have copied to you, so you can just subscribe to our channel. Thanks for all audience. Thank you, Dr. Karen Share, and
Yeah.
see you next MGBS.
Yeah.
Goodbye.
Thank you. Thank you. Bye, everyone. Yeah.